So our next speaker is James Jones from upriver at Stanford. We'll talk on contact networks, models of infection disease and simulating epidemics.
Okay, so everyone's been so well behaved and has stayed at the podium, and I'm like physically incapable of standing behind the podium.
Good.
So I apologize to the gentleman in the back room. This funny picture here. This talk is based in part on a presentation I gave down in Argentina this summer at a group that was convened by Josh Rosenthal of the Fogarty Center to bring social scientists and epidemiologists and ecologists together to think about how we can sort of do better with emerging infectious disease. And we got trapped by the Chilean volcano. So this is a picture of the Chilean volcano, and I just liked the picture, so I kept it as the background for my title. This was, yeah, in June. So this says, and I was thinking, you know, I got trapped in Buenos Aires for 5 days, and I was thinking the whole time, no, exactly, I was like, wow, I could be in Arusha, or I could be in Pontianak, and Buenos Aires is all right, you know, if I'm going to be trapped. More Malbec, please, you know. All right, so I always put my acknowledgments first because otherwise I often forget them. Much of the work I'm going to talk to you about today is with Marcel Slaté, who is a postdoc at Stanford and is an amazing assistant professor at Penn State now. And a bunch of the work is with other people too. So I am trained as a primatologist. I'm a biological anthropologist, and I was initially interested in chimpanzee and great ape behavior and ecology in general. And I don't think there are many events where you can really remember the exact moment that you had a transition, a professional transition, a life transition. Maybe you remember when you first saw the love of your life and you knew that everything was different. Well, I was sitting in the basement of William James Hall at Harvard. I was a teaching fellow for Richard Wrangham's primate social behavior class, and he was actually showing a slightly racier picture than this of some baboons. And he was talking about the dominance hierarchies. And for whatever reason, I had just read a little bit of graph theory. And I thought to myself, you know, I bet you could represent these primate dominance hierarchies as graphs, and maybe you'd actually get something interesting out of that. And this is when I sort of began my on and off love affair with social networks, and particularly their relevance to ecological processes and infectious disease in general. So here's the way we roll at Stanford. We do naturalistic observation. I'm not an experimentalist. I'm an ethologist, right? I observe naturalistic populations as Ms. Goodall of the apes here, now Dr. Goodall of the apes, Jane Goodall at Gombe, or this is what we maybe somewhat pretentiously call technologically enhanced ethological observation that I'll talk to you about briefly. So we gather data in naturalistic settings. We represent these data frequently using graph theoretic formalism, sometimes other ways. We use mathematical and computational models that use the social structure, and this is sort of the key, is that we really do care about the social structure and the ecology. And this is just a model for 2 viruses that infect colobus monkeys. And then, you know, we look at outcomes, and we try to make pretty pictures so that you have eye candy when you give presentations. So this is our thing, and I'm going to talk to you a little bit about. I'm going to talk to you about all of these things actually today. The standard model for an epidemic, of course, is based on a chemical kinetics model. You know that there's some supreme being who's pouring susceptible and infectious people into the giant Erlenmeyer flask and swirling them around, and you have a rate constant. And this always strikes undergraduates as a complete absurdity. Of course, it works pretty well, and ultimately I'm a pragmatist, and so sometimes it really does work if you have diseases that are easily transmitted through casual contact. And this is actually one of the outcomes of one of the projects I'm going to talk to you about today. Sometimes it doesn't work. Certain things are not easily transmitted through casual contact. And this is when we really need more explicit models of social contact. And here's just a figure I ripped off from Jimmy Adams. I shudder to think how long it took him to make this. But you know, you have a sexual network, and you have a bunch of people colored in red who are HIV positive, say, and you've got these concurrent partnerships. Everybody has two partners here, or at least all the men do, or all the whatever that is. And you think, oh, they've all got about the same risk because they all have a concurrent partnership. Until you look at the larger structure and you realize, oh my God, you know, there's a giant component here where I'm essentially connected, if indirectly, to an entire population of people through their sexual activity. And that all of a sudden you're thinking that these folks down here are doing okay. So this is what graphs and what networks have to offer epidemiology. And I'm not a graph theory zealot or a network zealot. I actually think there are lots of cases where the Erlenmeyer flask works perfectly well. Okay, so we'll transition through here. So I'm going to talk about, it's an ambitious talk. Yep, go.
Did you say that was an actual, that network came from, was an actual network?
Yeah, that's from Colorado Springs. Yeah.
Where? Colorado Springs?
Colorado Springs, yep.
At a school or a community?
No, it's a high risk sexual and drug transmission network.
Air Force Academy?
It's not. No. Carter can tell you all about this data set. So. Yeah. So as I say, it's ambitious. I want to talk about three vaguely related topics. The first, I'm going to talk about some work that Marcel and I have done on graph theoretic insights into optimal vaccination policies, or at least smart vaccination policies. I actually don't know if we can find an optimal vaccination policy, but sometimes we can find a smarter vaccination policy using our measurements of social structure. A second is the empirical measurement of contact networks. This is something that Marcel just got an enormous grant from CDC for, and we're moving ahead with this and pretty excited about it. And then finally, the thing that I'm actually most excited about right now is some work that I started with Beatrice Hahn, who's at the University of Pennsylvania Medical School, and has transitioned now. We just got an ecology of infectious disease grant to explore this more, to look at the simian immunodeficiency virus and the early evolution of HIV-1. So let's hope I can get to that. So the basic question we ask is, does social structure affect epidemic outcomes? And I'm going to whip through this. There's lots of sort of preconditioning that I do on this, and asking, you know, reviewing the literature, and all the social scientists say, well, of course it does. But you're often surprised when you read the epidemiological literature and you don't see a whole lot of work on this. I'm going to tell you that there are three basic flavors of network that we can consider here in very broad brushstrokes. One is a random graph, and all of these networks have the same mean contact. And one is a regular graph that you know, where everybody has, it's essentially a lattice. My lattice folded a little bit when I tried to render the graphic, but you know, it's essentially a lattice. And then the third is what we call well, it's a graph that has a lot of community structure to it. It has a lot of substructuring to it. And I posit three different types of social systems here.
Why are you doing that? We need sexual networks that look like any of those three.
Yeah, this isn't about sexual networks. Right. Yeah. Now,
so when you say networks, you mean certain subclasses of them?
I do. I mean, I'm now thinking more in a influenza type world here.
Right, but I mean, we should always keep that scope condition in mind.
Absolutely, and that's probably one of the slides I took out, because I go through and I show a sexual network that looks very different than any of these. It looks like the one that I showed before. They frequently do. They get these big long rings. They call them dendritic structures, right? That happen because of a very simple social rule, which is that you don't have sex with your friend's friends, right? And by avoiding having sex with you say your girlfriend's best friend, right? You explode out your networks, which is just sort of the opposite of many of these more casual social processes like the rest of life, right? With this community structure. So, thank you. Yeah, like I said, I stripped away so much information so that I could give 3 talks instead of 1. And then I've got Carter in the room. So this is, here's a, what may be a random, I mean, it's actually really hard to find a social process that really corresponds to a random graph. This is a picture from the Rally to Restore Sanity and/or Fear in Washington, D.C., where people are sort of more or less free to move about and mill around, and they have lots of face-to-face interactions. Here's a physical education class in China where everybody is unbelievably regimented in this enormous schoolyard. And here's a cocktail party where you get the natural formation of fairly intimate groups in a larger population. And I think that this is actually a pretty good model for lots of types of human social interaction. We live in households. Our households are organized into towns. Our towns are organized into regions. We have hierarchical structure to our lives. Most network epidemiology studies have focused on what's called the degree distribution, which is just the number of contacts you have. And there's been a huge amount of attention. And the last time I was at Irvine, I talked, maybe I ranted about this, I don't know. Let's see, there's been a great deal of attention given to degree distributions, and the results typically involve some sort of percolation theoretic extension of classical SIR models, where the results come from, and it turns out that many of the results that come from the so-called new network science are actually anticipated very well by classical ODE models that involve heterogeneity. Just saying. Less work is focused on the microstructure, okay? So, and this isn't a really good term because, of course, degree is kind of a microstructure, but by this I mean a sort of a higher order local structure, okay? So, for example, Martina Morris and Miriam Kretschmar have measured concurrency based on a line graph of the adjacency matrix. And it's really a measure of betweenness. So betweenness is you have 2 individuals, and the question is, do you have a bridging person in between them? This is betweenness. Networks with this higher betweenness are more centralized and can yield larger epidemics. In Martina and Miriam's analysis. Assortative mixing is another thing that's been looked at quite a bit, and both by age, by sexual activity level, so by mixing by attributes, or mixing by degree. And these can affect the likelihood of an epidemic in complicated ways, depending on what the type of mixing is. Mark Hancock and I showed that for a fixed power law degree distribution, you can have lots of, the propensity for short paths, basically meaning you're clustering, can pretty radically change the overall epidemic likelihood, even though the marginal degree distributions are identical. So this was sort of the thing that got me to start thinking about this. We did this in 2003 before we really had a very good way of doing it, and I've wanted to get back to it for a while. So this is something that Marcel and I took up. And the key point I want to take away from this is that a huge amount of this work on degree distribution confounds activity with structure. So if you have a power law degree distribution, okay, so that means that most people have few contacts, a few have a great number of them. You're going to have a lot of stars. And those stars are obviously the individual who's at the center of a star has a high degree, but that person also has a high betweenness or whatever other measure of centrality you want to look at for the graph, because in order to get from this side of the graph to this side of the graph, you got to go through the hub. Okay? So this is the basic idea that we built on, is let's see if we can come up with graphs that we simulate that are uniform in their degree but vary in their structure, right? So here's just a quick just example of what happens to betweenness. Here's a star, right? And you've got one individual who has or central. I shouldn't say betweenness. I do mean just centrality. This is a highly centralized graph, right? This guy has whatever it is, 10 contacts. We then take two stars and we link them, right? And once again, we have these highly central individuals, but then we have these linking individuals who are even higher, right? And so you get this you start to form a distribution of betweenness. When you have two linked clusters with more distributed within cluster connections, some people have a few more. They're on a few more paths, short paths in here, but still, these bridges really dominate your distribution of betweenness. And this is a nice example of how your brain. We're not really good at actually detecting structure in graphs because these are two highly clustered subgraphs that are linked together. And I don't know, maybe Carter or Katie can see that, but most people can't. There isn't much. It doesn't look like there's much structure. There actually is if you measure it. But there's much more a sort of widely distributed distribution of centrality here. And so one could imagine in a situation like this, you can see a lever for control. Like say you had an outbreak over here, if you could, shut down the road out of town, as it were, right? You could prevent a little outbreak here from traversing across to this subgroup. Okay, and so the question is, can we be clever about it and find the roads out of town in this situation where it isn't so obvious that there's a single path out? Okay. So what we did was we simulated, reasonably sizable networks with different levels of community structure, meaning this propensity to form within group versus between group clusters. We measured this using Girvin and Newman's Q statistic. We control for heterogeneity. All our networks, everyone has the exact same degree. They all have 10 connections. The only thing that changes is whether or not those 10 connections fall within a local cluster or more globally in the network. And then we simulate SIR epidemics on these networks with the networks as a substrate. And it's important to note that they're SIR epidemics. All these results are predicated on the fact that people can get better.
Jimmy, why do you fix the number of connections? It seems like a curious word choice to say that you control for heterogeneity, right?
Contact heterogeneity, right? Because I mean, what we know, starting with Anderson et al. in 1986, we know that heterogeneity in contact number, has a driving force on epidemics, that the basic reproduction number is proportional to the variance or to the coefficient of variation. And so we wanted to just take that entirely out of the...
No, I understand that, but so you're making a world in which heterogeneity doesn't matter because there is none.
The only heterogeneity is in the group structure, yeah.
So that can partial out the effect of group structure, but it doesn't necessarily mean that back in the real world where there is heterogeneity. Right?
Am I talking about the real world?
I don't know.
This is theory. This is a little bit of theory.
As rule of order, we're not allowed to bring the real world.
Okay.
So disturbing to draw such a bright line between theory and the real world, though.
No, I mean this.
We hope to do
This
something.
is a question of can we isolate just this sort of mesostructure, right? That structure versus activity. I'm sorry?
Regular random graph, just a regular but random, right?
Yeah. Well, no, actually, so I'm sorry. The way we start off is it starts off lattice-like, and then we rewire everything.
So then it becomes irregular.
Yeah. Okay, and we measure we measure a bunch of epidemic metrics, like in particular final size, which is the fraction of all nodes in your network who are infected by the end of the epidemic. And we evaluate the effects of community structure. And here is all of this paper and everything condensed down into a single figure, where we have this measure of modularity, the Gervin and Newman's Q measure. And this is a really amazing figure because there's this almost impulse-like quality to it. So this is for 3 different levels of transmission. And well, you notice that it's most important for high levels of transmission that you start from a completely modularized graph where there's essentially one, so they're all connected, right? They're strongly connected graphs, but they're connected only by, at the highest modularity, by a single tie between groups, right? That each subgroup has only one road out of town, okay? And then you break that clustering up as you move down here, and you make this fairly abrupt transition right around 0.92, 0.93, where you go from having very small localized epidemics to having essentially as big an epidemic as you're ever going to have. Remember, there are 2,000 nodes. And I was pretty surprised by this, because what happens is that here, when you're really clustered, you have your SIR epidemic, it's bouncing around in your local community, and eventually it just runs out of fuel, and it hasn't gotten out of town in time before you have stochastic fade out, okay? And as you open up more roads out of each town, you greatly increase the chance that it's going to escape from the local cluster and make it into the broader graph. And when it does that into enough local clusters, then you have a full-blown epidemic. So the simple answer, does social structure have an effect, independent of just activity level, I think is a pretty resounding yes. Now, and here's a nice example. This is an epidemic curve of a not very heavily clustered graph, and it looks pretty much like we'd expect an epidemic curve to look like. And here is an epidemic curve for a highly clustered graph that actually had a big outbreak. We had to go and look for it in these hundreds of thousands of simulation runs. We had to find one. And what does it look like? Well, it actually looks like you've got a series of little mini epidemics. You go into a community, you get your epidemic curve, you're starting to go extinct, and then from the virus's perspective, you get lucky and you get out of town. You start another epidemic. And then there's another. So what we have here is, this gives you a sense of why it's so improbable. We have these serial small epidemics.
Can you remind us what the colors?
Oh, the colors just indicate the generation of transmission. So red here is you're infected by the index case, and then on down the line. Okay. As I mentioned, centrality in general, betweenness in particular, and degree are highly correlated in lots of different graphs. And it turns out that as you, let me think for a second, so as you get more and more highly clustered, right, you see as Q goes up from 0.77, we've got a very close correlation with an R-square of 0.839 between centrality and degree of individuals through the network. This breaks down as you get more clustered, and as you get highly clustered, you basically break the connection between your degree and your betweenness. Yep.
I thought these were regular graphs.
This is not a regular graph. This is.
So what is this one from?
This is a simulation with the same mean degree, but now you've got it's essentially a Poisson variation.
Okay, so these are just, this is what you see on the random graph.
Yeah.
Beautiful.
Yeah.
Okay. Want to clarify.
Yeah. All right. So this led us to ask the question: can we actually use this information to develop a smart vaccination study? And we found that. So here are the various. There's no way I'm going to get through all three of these. Here are the various strategies we used. I'm going to zip through them. Random. You vaccinate a fraction p of your graph. You go in and you count up everybody's degree, and you vaccinate by the highest degree on down until you vaccinate a fraction p. You calculate everyone's betweenness. This sounds really realistic, doesn't it? And you vaccinate it in descending order until you reach a fraction p. Now even more realistic, right? You calculate the random walk centrality, which is of course a hugely costly calculation. And you again vaccinate until you reach a fraction p. Then we get stupid about it. We say we vaccinate people's acquaintances. We go and we say, all right, name a friend, and they say, well, my friend's Joe. You go and you vaccinate Joe, right? And this is sometimes known as the Cohen algorithm. And the idea is that your friends are on average more highly connected than you are, and so that if you vaccinate an acquaintance, you're actually getting more highly connected individuals in the graph. And it works amazingly well, particularly if you have power law degree distributions. And then we use an algorithm that we developed called the Community Bridge Finder. And I'm going to focus, not exclusively, I'm going to focus primarily on these two because they only rely on the type of information that one could plausibly gather during epidemiologic investigation. They rely simply on contact tracing, information that is sometimes gathered in outbreaks. So they're local. Okay, so the logic of acquaintance vaccination is that a star has a central actor. If you sample randomly, you have a 1 in 11 chance of picking any given actor, and you have a 10 in 10 chance of picking the central actor when you ask them to name someone who's an acquaintance. Okay, and so you greatly increase the average connectedness. Right, this is an absurd case, but it works on average in general in a network where you have a lot of stars in particular. Here's the Community Bridge Finder. You start with an infected individual, ego, and you say, and so I'll mention that this works only and we're working on a paper that actually describes the Bridge Finder algorithm better. It works when you have an emerging infectious disease, when you have low potential vaccination coverage, and when you have a community graph, where you have this clustering. It doesn't work for sexually transmitted diseases, and as you'll soon see why. So you have an outbreak, you have an index case, and you ask this person, name your contacts. You then randomly choose one of their contacts, and you go and you interview this person, and you say, okay, you get all of their contacts, and then you randomly choose one of their contacts. Okay. Now you ask the question, do you know ego? You of the second wave of contact tracing. And if you don't, if this person doesn't know our focal individual, then we do another round. So we're doing this random walk on the graph. Okay? And then you ask, do you know ego? And you might as well ask, do you know the first generation too, right? Because it's an easy question to ask. And if they do know them, if they do know ego, you keep going again. And you just keep going until you reach someone who doesn't know ego. If not, you vaccinate this person and all of his or her direct contacts because you've found a bridge between two clusters. Okay, you've come from here, you've gone to here, to here, to here, doesn't know ego. Probabilistically, this person is in a different cluster. Okay, so we can now implement this in our simulations and see how we do. And for this, we used they're kind of like data. we have Facebook information, which we then sort of trim down a little bit because we have Facebook friends, and this is back from 2005 when Facebook was actually like a college thing. We have it for whatever it is, 6 universities. And we reduced it down to make them more like actual contacts by you were a contact if you were Facebook friends and you either shared a major, a dorm, or a year in college. And all of these plots, if there's a red bar that goes above, it means that our algorithm beat the alternatives at a given level of vaccination coverage. And you'll see that these are low, right? They're low. This is outbreak control. This is not like influenza control or something, or childhood diseases. So and here, you know, we can then alter we can take the data. We can rearrange some of the connections. We can change the modularity a little bit. And again, and I'm just very quickly summarizing the random walk. This is if you have complete information. Random walk is best. Betweenness is next. Degree is next. Right? That this is the order of preference for what does the best job for vaccinating in these Facebook networks. And then if you don't have that information, well, the random walk always does better, but the community bridge finder does better than the acquaintance method. And yep.
The way you coded that, saturated blue is right next to saturated red, right? Which kind of does a number on the visual.
Does it? Yeah.
Well, because saturated blue and saturated red are actually very close to each other, right? So then white is most different. I mean, it's kind of hard to read that graphic, right?
Yeah.
So those, well, we can talk about it later.
Yeah, good, because I really want to get to chimps.
Okay.
Please. It's also faster, right? It's a more efficient algorithm than the more modified version of the Cohen algorithm, which is more involved, that involves multiple iterations. No, I know, it's hard to beat, name your friend, my friend's Joe, vaccinate Joe. No, it's true. That what they actually do in their paper is something considerably more involved. Okay, so, and just to summarize this, in these small world, these community networks, Betweenness does the best, and well, actually, this shouldn't be this should be random walk Betweenness. I mean, it's not strict Betweenness. It's random walk Betweenness does by far the best, but our community bridge finder does next best, and so we like that. Okay, but I used Facebook data, or we used Facebook data in these simulations, and that was painful, right? And why did we do that? Because there are no network data for the types of infectious diseases that we're imagining, community-based diseases that may be transmitted through droplet or aerosol or something. There are some very hard-fought data for sexually transmitted infections, the Colorado Springs data, for example, but there's so little for face-to-face interactions. And so Marcel brought this proposal to Stanford, and we got an NSF rapid grant to buy these to buy the moats that allowed us to do the measurement of contact networks within a school. So the network epidemiology is hampered by the absence of detailed empirical data. Facebook, I'm shamefaced about it, right? But the goal is to measure detailed close proximity interaction data in schools. And we use this technologically enhanced ethological observation. This is what's called it's a Telos B. It's a wireless radio device. It's a little smaller than this. We have students wear them around their necks on a lanyard. It does three things. It emits a weak radio signal, it logs radio signals, and it writes them to volatile media. It's They could be way, 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 way smaller than something like you'd wear around your neck, but it turns out they're hampered by the power source. It runs on AA batteries. So you might as well not make the circuit board too small, because you got to put the AA batteries in there anyways. I took out the slide, I shouldn't have. We're extremely lucky to have at Stanford in the computer science department the guy who wrote the operating system that these things run on, called TinyOS. And so Phil Levis in the computer science department is a collaborator with us on this, as is Mark Feldman in the biology department. It's a nice thing about being at Stanford. So I'm going to just cut right to the chase. We did this at a large school in the Bay Area. We did it for one day, and it was an enormous logistical hassle. It was kind of a nightmare, and thank God Marcel is like the hardest working, most Swiss, most dedicated person you've ever met, because he made this happen, and he's got the new grant to keep measuring this over multiple days, which he can hire some more people, hopefully, who are half as hardworking as he is. But we have 2.7 million unique close proximity interactions in a day, in about 1,000 motes that were deployed. We've got all sorts of fun sort of, you can see the flows between classes. You can see the and the sampling period for this is every 20 seconds. They ping, right? So we can measure contact networks down to 1/3 of a minute, and we can measure them up to whatever sort of grain we want to by aggregating consecutive logs. So here's some degree distributions. And what we see here is that we've got the number of unique contacts. They're colored by, so the blue are students, the purple are teachers, the green are staff, and the red are other, which means actually mostly our people we had some grad students who were there, and Marcel. I'm guessing Marcel was the guy who's out here who had a lot of contacts, because he was no doubt running around like a madman trying to make sure that everything was working. And what I want to draw your attention to is if we go over to any of these plots and we just look at the students, we see that the number of interactions is actually remarkably Gaussian looking. Okay? And this gets back to this idea of power law distributions of networks. We often assume that they're power laws because, well, isn't everything a power law? And I don't think it is. Here's a fit using the sort of standard plot that gives me the willies when I see it. And you can just see that it's a bad fit, right? And if you don't believe that, here's the way it seems like you should plot it as a histogram, as probability mass with a fit to it. And this is a Yule distribution, a power law. And this is actually a Gaussian fit, and it actually fits very nicely.
But if you were a physicist, that would have been a power law.
And if you don't believe me, I did a Kolmogorov-Smirnov two-sample test just to show that the power law does not in fact fit very well. And actually, I've fit now quite a few models using maximum likelihood to these data. And in fact, the normal distribution fits the best of everything that I've tried so far. Now, you'll notice that there is a spike right here. This is an artifact that Marcel has just discovered. And what actually fits the best is a 2-component normal mixture with essentially a 0 variance, essentially a spike right at the middle. And now we figured out that there was an artifact in the data. And in fact, a single Gaussian fits very well to the student data. And a nice mixture fits very well to the aggregate. Right? And a number of people have said, well, geez, I mean, it's a school, these students are constrained, they're in classrooms, they're not allowed freedom of movement. I'm like, welcome to life. It's called sociology, right? Social structure constrains us, right? Where we live and where we work and who our boss is and who our teacher is, it affects the types of epidemiologic contacts we can have in addition to lots of other things, right? So obviously it would be very interesting to scale this up to looking at, say, China, right? But it's not going to happen because it is such a logistical hassle to even do it in a school, in a highly regulated environment. There are certain aspects of China that would make it easier to do. And just a relative distribution plot, yep, it really does work. There's the empirical CDF. And
how did you calibrate your signal cutoff for your modes?
it's 2 meters. Yep. And the idea being, and you know, as with your pen and paper data collection, you know, face-to-face contact, these things give off such a weak radio signal that if you know, if I'm facing Andrew, and we had motes on, we'd ping right now. But if I were to turn like this, we probably wouldn't ping, right? And so, you know, we see this as the radius of sneezing, or, you know, of ejecting droplets into the air. And so I like the way you restrict your data.
So did you test that? Did you test
We did.
the face-to-face?
Yeah.
It would be cool to do like a graph of the, because it's not a
They've, no, I know, yeah. They've done it. And I'm a co-author on the paper. It's in a computer science proceedings, but Phil's grad student Maria and Marcel have written a paper that does all this stuff.
Do you know which proceedings?
I do. I can give you the reference. Yeah. Yeah.
And to be defined as a contact, did you have to be in did they have to think for a certain period of time? Or was it just any signal pickup?
It's funny you should ask that. I'm going to go right to this, which is we can affect the sampling rate. We could say that a contact is one single line in the log of the log file for each individual. Or we can say, well, you know, those are really transient contacts. We define our contact as being 3 or more consecutive or 5 or more consecutive, right? And we can do that. And we can construct graphs based on these different sampling rates. And I'm just going to, this is a whole can of worms that I talked to Carter about and Katie about last night. But what we can do is we can take our graphs from these constructed from different sampling rates, where you require, you know, greater and greater number of consecutive logs between 2 individuals to define a contact. We can then construct these graphs. We can then run simulations on them, and we know who our seed is for each epidemic simulation. And we know from our past experience that the final size is well predicted by things like that individual's degree and that individual's betweenness, okay? So what we can do is then look at the R-square. When we do this 1,000 times, we can look at the R-square between the seed's degree or betweenness and the final size of the simulated epidemic as more or less a measure of, you know, what we think, you know, how effective the graph measurement of social structure is. And what you can see is that the maximum correlation, right, between these individual level attributes and the final size of the epidemic on a graph of that particular scale happens at 45 minutes.
What was the length of the average class period?
Yeah, they're an hour. Yeah. So this is where I come back to, you know, remember the picture of Jane Goodall with the binoculars? I'm like, you know, that looks like you could do scan samples with pen and paper, you know, and look at who has, you know, you have a well-defined criterion and you can just, you know.
But now technically though, what you're doing is not really a sampling rate, right? You're not saying I'm going to at 45-minute intervals take a cross-section.
You're right.
You're saying actually this is the
Yes,
duration threshold.
That's right.
So that would be a better term for it.
Yeah.
Because you could have people with, you know, you go
That's right.
for every 45 minutes and take a slice, and
Yep.
a lot of those might not be.
Right, so you wouldn't want to take a scan sample at every 45 minutes, right? You'd have a more complicated criterion for contact. But I think that there's something to be said for, you know, you don't have to go to motes to get high quality contact data. And I think that that's actually a really important outcome of this, because You know, I know that CDC is very excited about moats right now as a way of measuring contacts, and I think there are real costs to them. That for targeted types of questions, they can work really well. But they're not a panacea for measuring contacts, and I think we can do an awful lot with good old-fashioned behavioral science. So we've got highly detailed measurements. They're very effective at measuring interactions in a school context, but they're highly labor-intensive, and they're somewhat disruptive, right? That, you know, you have to rely on the students becoming a little bit habituated. We went in multiple times. Phil and I gave lectures to the entire class. Marcel was going into the science club weekly. This was a major endeavor. We had to get the school on board. We had to get all the teachers on board. And it's a bit disruptive to their day. Contacts are not a power law, that they're actually simple mixtures of Gaussians. That graph measures dependent upon temporal scale of network sampling. So I didn't show you all this, but we can do all the graph theoretic calculations of the networks. And it really matters what the temporal scale is. Big surprise, right? And extremely high frequency sampling may not actually be worth the effort for flu. And then I didn't talk about vaccination. It turns out that you can't actually do a whole lot better than random vaccination. You can do a little bit better, but there's no like obvious structure to the graph that allows you to say, we need to fit, these are the high elasticity individuals. Just vaccinate a fraction p, you're good. And then there seems to be an assumption that degree distributions of all social networks have long and or fat tails, and that it arises from socio-technical bias in existing network databases. There's no cost to, except when you friend your mother, maybe. There's no cost to being a friend on Facebook. You just click, right? Real relationships, not only are they constrained by our social structures and our geographical structures and all sorts of other things, but they also carry costs. You have to maintain relationships.
But Facebook's not a power law either. People claim that that's because they sample it, right?
Yeah.
If you sample it correctly, you see Facebook isn't a power law. I'm not sure that anyone has shown an actual social network of actual individual people that is a power law. There may be some, but most things that are tend to be power laws aren't.
Yeah.
If you look at even if their own plots, they're clearly not. And then often things that are claimed to have bad sampling problems.
Yeah.
So I wouldn't necessarily put this at the field of socio-technical bias.
Yeah.
And so the small days, that's often don't look even big like it. They tend to be heavy-tailed.
Yeah.
But it's just that's not there.
All right, so I think the real takeaway, though, is to think hard about social process. Actually try to understand how people form friendships, how people form sexual partnerships, how people, make casual contacts, and that you sometimes can model those things, and that you can actually get real insight, sometimes even analytic insight, into the properties of these systems. Okay, the last thing I want to talk to you about, Carl, how much time do I have? 5 minutes. All right, good, easy. Is SIV and the early evolution of HIV. In 2001, I started collaborating with people at the Jane Goodall Institute. This is Jane, 1964, taken by her then husband, Hugo van Lauwek. Jane's been doing work at Gombe National Park in Tanzania since 1960, and there it is. It's on the shores of Lake Tanganyika and in far western Tanzania. It's a tiny little sliver of park. These are different groups. In 1972 and 2008, we're down to 3 groups in Gombe, and Jane's original habituated group is this green one, Kasekela, and all the data that I show you will be from Kasekela, and with a few now from Mitumba. So vital event data was first systematically gathered in 1964, so we have 46 years of recorded births, deaths, and transfers in a community of known individuals. We haven't entered the last year yet, which is why we're a year short. The chimpanzee strain of the simian immunodeficiency virus was first detected in Gombe in 2001. Beatrice Hahn, whom I've already mentioned, and her colleagues developed a non-invasive test for SIV from chimpanzee feces about this time. And feces have been genetically fingerprinted to identify the pooper, which is great. So you can just pick up poop in the forest. You can pull out the SIV, and you can pull out the identity of the person who left it, or the chimp who left it, without actually having to see because no one wants to watch a chimp poop. So we've been monitoring the epidemiology continuously since 2002. SIV prevalence has risen and it's declined a little bit since then. And so this is a paper that, this is Beatrice's postdoc, Brandon Keel and myself and Rebecca Rudisell and you know, a whole group of people wrote in Nature in 2009. It peaked about 2006. It's been declining ever since. And we'll cut to the chase that we were able to code chimps as being SIV positive or SIV negative. The received dogma is that SIV doesn't kill its natural host and that HIV is so devastatingly pathogenic because of a recent spillover. It's not well adapted to its new host. This turns out to be utter rubbish. That in fact, SIV, so this is a discrete survivorship curve. The proportion surviving from birth until age out here on the x-axis, and the SIV-infected chimps die at a much higher rate, and particularly, you know, there are not a lot of events here, but there actually are a substantial number of chimp years, right, when you actually analyze it by the chimp year using discrete event history analysis. There are a couple different ways you can actually categorize some of the deaths. 2 of the infants died, and we weren't able to actually extract virus from them because we didn't have fecal samples from them. They had antibodies. They could have been maternally derived antibodies. And so, but even if we redefine them as not SIV positive, and we redefine a couple exposure years for animals who are somewhat ambiguous, it doesn't matter. And we made the ephemerally. You know, this is the problem. There's no more like, you know, there's no more Dewey wins moment when you hold up the newspaper that's shown even a right, even a correct headline. If you want to get the picture of your study when it makes the New York Times, you better take a PDF shot of the web page, which I happen to do. And there we are. We made the New York Times if ephemerally. It didn't last very long.
Don't feel bad. I got kicked off the front page of Time by Snooki.
We've got our priorities as a society, Andrew. You know, we can construct sexual networks. So I should just say that depending on how you do it, that 9 to 16 times the hazard ratio, right? So that there's a real hazard to having SIV as a wild chimp. We can construct sexual networks. We've observed a certain fraction of all matings among the chimps at Gombe. Whenever females only mate when they're in estrus, and whenever a female comes into estrus, she's followed constantly. I know. Well, they're habituated. They don't seem to mind. And so we capture quite a few of the total mating events. Every once in a while, they'll run away to the bushes, and it's hard to see. But here's a sexual network of all the observed matings in a single year, where the nodes and the edges are colored red if it's an SIV positive chimp. And again, I challenge you to find some structure to this. I don't see much. Chimps have what's called a promiscuous mating system. It's a technical term, and it means that a female will attempt to mate with every single male in the group whenever she's in estrus, and all the males are perfectly willing to help out with this goal because they find this enormously swollen bottom of the female chimp just unbelievably attractive. Okay, and so talk about a system that's like designed for the spread of a sexually transmitted retrovirus. And yet we only have 15% prevalence at max. Here's 2006. You know, it looks more or less the same. We can use these data to try to calculate R0 based on a variety of assumptions. And here's our observed. We take the number of matings of males and the number of matings observed in females. And we can put together an estimate that puts us at about, this is between 2 and 2.5. Those are those isoclines there. So, you know, there's a pretty high R0. We expect this to be a pretty vigorous epidemic. We see all this contact. This is what's driving R0. And so why are there still chimps at Gombe when it kills them at 16 times the baseline rate? Carl's standing, so I have to hurry. So SIV increases the hazard, 9 to 15 times. HIV-1 increases the hazard. It's actually hard to measure this, but you can go back to some of the earlier literature, find it increases the hazard ratio about 18 to 60 times. HIV-2 between 2 and 5 times. So SIVcpz is intermediate between HIV-2 and HIV-1. Has a very high R0. It has, just explosive, it's seemingly explosive potential for transmission within the group. The sooty mangabey flavor of SIV does not kill sooty mangabes. So I don't know what that says, but that's an interesting point. The thing that's important, I'll take like 2 more minutes, okay? 1.5, okay, is that we expect that this level of mortality should really induce population decline and probably extinction in these chimps. Here's a plot that's just, this is the critical threshold below which you get extinction, right? And here's the mortality multiplier for 3 different levels of fertility multipliers. And what we see is that our 95% confidence interval for the mortality multiplier is here. This population can't stand any SIV apparently and persist over the long run. So we developed some stochastic models to look at this because they are small populations. And what we found is, and as soon as I saw it, I was like, oh God, I'm so dumb. How could I not have realized that? Sure, chimps go extinct a lot, right, in these populations. But you know what? So does the virus. I'm like, oh, all right, the virus goes extinct. Because it's so pathogenic, it burns out all its fuel very quickly, and because it's a sexually transmitted disease, you basically have half the effective population size for transmission. 80% of the time in these simulations, the virus goes extinct before the chimps do. And so this raises lots of questions about the early evolution of HIV. When it spills over into human populations, you're in, it happens in West Central Africa. You have widely distributed villages that are weakly connected through migration and trade. How is it that HIV managed to stick this one time when some cut hunter got an initial infection of SIVcpz? And so this is the question that motivates a new project that we're doing in the Kibale Forest. We just got an Ecology of Infectious Disease grant to do this, where we've got a model system of red colobus monkeys who are connected in a metapopulation that looks like this, right? A population of populations. They're infected with SIV-CoL. Some populations don't have it, some do. They're linked through dispersal and occasional agonistic interactions between groups. And so we're going to measure transmission. We're going to measure the fitness consequences of infection with SIV and 2 other retroviruses. And we're going to use this as a model system for trying to understand the early evolution of HIV-1, which is filled with all sorts of colorful and tragic characters and interesting bits. And I'll just show you a couple figures to whet your palate for this. There's a network explanation. A metapopulation is just a type of network. And smallpox vaccination may be implicated, and so might sex, because these are frontier towns, and frontier towns have problems that they bring with them. I'll end with this, that this was a saying in the Belgian Congo that it's a play on words in the French. They've brought us civilization. They've brought us syphilization. Syphilis rates were astronomical. I mean, an incidence of 10% of syphilis in Congo in about the time that HIV emerged. Anyways, that's another lecture, another talk. I'll stop there.
So is SIV probably isn't as lethal as HIV?
Well, I mean, it seems to be intermediate between HIV, you know, and this is terrible systematics, right? We call these two independent viruses that are clustered in clades of things we call SIV. We call them distinctly HIV, but we call one HIV1 and one HIV2. They have distinct evolutionary histories. So the
This is where understanding the internal dynamics of a migrant would
Yep.
play a big role.
No, I think so, absolutely, yeah.
I've understood that the role of primary infection, acute infection, is huge in
I think it is huge, and I think that that's the only way you can understand why these chimps, given that basically every female mates with every single male, including all the SIV positive ones, and all the SIV positive females mate with all the SIV negative males, that if you're only infectious during primary infection, then that could explain how we get these networks. Yeah. And so this is a current modeling focus for our work.
Mine too.
Yeah, well, good.